All right, team. Today we're going to talk a little bit about what controls when and how much of an RNA is made. Now, in other videos, we've talked about promoters and we've talked about transcription factors, as well as consensus sequences, that is, pieces of DNA that transcription factors can bind to, and often these consensus sequences are found in the promoters. Altogether, these three can combine to help determine when a gene is turned on, that is when transcription is occurring, and how much that gene is turned on, how much transcription is occurring. And this is a phenomenon that occurs both in eukaryotic cells, so in humans, in insects, in yeast, in plants. All eukaryotes, multicellular organisms, and single-celled organisms have to regulate how much of a gene is turned on. And this is also true in prokaryotes, in bacteria. And today we're going to focus on a strategy that bacteria use combining these aspects of gene regulation, and that's called an operon. So let's talk more about operons. First, let's think about an operon. So we've thought in the past about genes having a promoter region and then a gene that is transcribed into an mRNA. That's exactly what an operon is. You can see we've got a promoter right here. And then as part of the promoter, we've got an operator. Really, this is just an example of a consensus sequence. And then what's different about an operon is rather than having a single gene next to the promoter, we've actually got 1, 2, 3 genes, or that is more than one gene lining up next to this promoter. So when we have transcription occur, we have transcription of a single mRNA, but you can see that this mRNA has more than one coding region, just like the DNA it came from, more than one coding region in both the mRNA and the DNA. And then ultimately, we end up translating more than one protein from this. So if we looked closely at the mRNA, here would be a start codon, here's a start codon, and there's a start codon. And we could also draw in, here's a stop codon, here's a stop codon, here's a stop codon. So we've got an mRNA that's translated into 3 separate proteins. Often these proteins work together to perform some sort of cellular function. So they often work together. Now, this isn't the only component of an operon. We need to have a regulatory protein that's part of this operon. And you can see over here we've got a regulatory protein that has its own promoter region and its own coding region. We have one gene that's transcribed into a single mRNA, and that mRNA is translated into a regulatory protein. So this is an example of a transcription factor. It's a DNA binding protein. And this protein can come over here and bind to the operator. Remember, that's a consensus sequence. It's going to look for its consensus sequence in the region of DNA, and it's going to bind to that DNA. And there are 2 types of regulatory proteins. This regulatory protein could be an activator. That means it turns on transcription or helps turn on transcription. Or a transcription factor could be an inhibitor. That means it blocks or prevents transcription. Either way, this regulatory protein will help determine when the operon is turned on and making its different component proteins. Let's talk a little bit more about how an operon can turn on gene expression. So we have two things to think about. First, the transcriptional state of the operon. That is it normally off or is it normally on? If the gene expression is normally off and it can be induced to turn on, we say we have inducible expression. Now, if the gene expression of that operon is normally turned on, but some signal can change it, repress it to turn off, then we have repressible operon. We can also have two types of control over that transcription. That is, what type of transcription factor are we dealing with? We could have a negative regulatory protein, that is a repressor or inhibitor that can turn off transcription or block transcription from occurring in the first place. We can also have a positive regulatory protein, an activator that helps turn on transcription. And you can have an activator that's involved in an inducible operon. You can have an activator that's involved in a repressible operon. And we're going to talk about all four combinations here. First, let's think about an inducible system. That means a system where gene expression is normally off, but we're turning it to on. So first I'm going to draw out the DNA here where we have a gene, but transcription from this gene is not turned on until something happens, something changes in the environment that induces gene expression to turn on. So we've still got our DNA here. Then we've got mRNA being transcribed and ultimately a protein being translated. The first type of inducible system we're going to talk about is a positive inducible system. And in this case, we're going to have a transcription factor that's an activator. So I'm going to draw a little activator protein here. It's got a plus because it's positive. And a positive transcription factor is going to be binding to the DNA when transcription is occurring. So is this positive transcription factor going to be binding here or here? Well, it's going to be binding down here because this is where transcription is occurring. So I'm going to draw my little positive transcription factor on the DNA. But of course, this transcription factor needs to be around. It needs to be hanging around up here, waiting to receive some signal to help it turn on gene expression. And what might that signal be? Well, we could say that salt or sodium is the signal for this transcription factor. When this transcription factor binds a sodium ion, it is able to change shape and then successfully bind to the DNA, allowing transcription to occur. And now we're going to turn on transcription of a gene or even a set of proteins. Remember, this is an operon. And maybe these proteins all help deal with salts in the environment. Okay, let's draw another inducible system. Here's our next inducible system, and in this case, we're going to be talking about a negative transcription factor or a repressor of transcription. So we're going to have a negative inducible system. And an example of this is metabolism of lactose or the lac operon. This is a very common operon that many bacteria have, and it's well studied in E. coli. And in this case, we're going to have a negative transcription factor. That negative transcription factor binds DNA when transcription is not occurring. It's negative. Okay, so that means we're going to have the transcription factor binding up here. And when the transcription factor, when this inhibitory transcription factor is bound, RNA polymerase is going to try, but it's not going to be able to succeed in doing transcription here. You can see transcription is blocked. But when some stimulus, some environmental signal is perceived by the cell, in this case, it's lactose, that's going to change the shape of the transcription factor. In this case, when our negative transcription factor binds lactose, it changes shape so that it falls off the DNA. Now RNA polymerase is ready to get on the DNA, allowing transcription to occur so we can make mRNA and then make our proteins that help us or help bacteria digest lactose. That's an inducible system. Let's talk next about a repressible system. If we're going to talk about a repressible operon, this is an operon that's normally on, and we're able to turn it off, right? We're able to repress it. So that means we normally have transcription occurring. We're making an mRNA here. We're transcribing it from the DNA. Since this is typically occurring in bacteria, we're going to be making some proteins. We're going to be translating just as soon as transcription occurs. But then something shifts in the environment and transcription turns off. Okay, that's a repressible system. And now we want to think about this repressible system with a positive transcription factor. A transcription factor whose job it is to help gene expression occur. So is that positive transcription factor going to be binding up here or up here? Well, if it's helping transcription occur, you can imagine that the positive transcription factor is going to be bound to the DNA right there where transcription is happening. We're getting production of the mRNA. Okay. And that positive transcription factor is going to help transcription until something changes in the environment so that the transcription factor falls off the DNA. Okay. Maybe we'd say in this case that we get a hormone signal, and that hormone tells us that we need to turn off gene expression. So now the transcription factor is going to bind to this hormone. It's no longer able, you can see it's changed shape a little bit here, so it's no longer able to bind to the DNA. And now it's no longer sending out the call to RNA polymerase to do transcription, so we end up blocking transcription without that helper transcription factor. Let's draw one more repressible operon, and we can talk about a negative system. All right, here's our new repressible operon. We need to think about how this repressible operon could be regulated by a negative transcription factor. That is a transcription factor that turns off gene expression. Now, is that transcription factor going to be binding up here or down here? Well, we know the positive one is helping gene expression occur. The negative transcription factor is going to be binding where DNA transcription, or transcription rather of RNA, is blocked. So up here in the positive situation, this transcription factor needs to be floating around, but it won't bind to the DNA until some external stimulus tells it it's time to bind. Okay, so this is a common example of this one is the tryptophan operon. So we need to be making these proteins that help us make the amino acid tryptophan. But when we build up enough tryptophan in our system, it can bind to this transcription factor and actually help it change shape so that it can bind to the DNA and say, let's turn off transcription. We've got enough tryptophan, there's no need to make more. Okay, so again, we've got our negative transcription factor floating around looking for this environmental signal, and when it's perceived, then we can get the tryptophan binding repressor protein to come in here. Now RNA polymerase is not able to do its job, so we don't have any transcription. And that's just what the cell needs because we have enough tryptophan, no need to make any more. All right, so I hope these examples have helped you think about repressible and inducible operons and how they can be regulated by different types of transcription factors.